of Montpellier. I started off as a biologist. I'm from Greece, originally from Athens, where I did my undergraduate degree in the south of Greece, in Crete, a big island, and I studied molecular biology. Then I left with an exchange project program that is to go to Ireland for a year, where I started bioinformatics. And then I moved over to Oxford University to do my PhD in molecular evolution, and more precisely in phylogenetic analysis of viral sequences, before moving over to the States, where I worked in Flybase. Flybase is a database, well, actually it's a knowledge base that treats all the information concerning the Drosophila species, not only Drosophila melanogaster, but for all the other species. They are several different teams that they are working on this project that is managed by the Harvard University. Then I moved to the private sector, working in the States and in Europe for different companies in the bioinformatics field. I set up my research team in the academia in Greece, working in comparative genomics projects. And since 2014, I'm in France, where I got a professorship at the University of Montpellier, heading the IMGT, the International Immunogenetics Information System, which is what we're going to discover together these 2 weeks, this week and the following week. So we are going to touch upon immunoinformatics, which is a specific field of bioinformatics. So basically, that's a little introduction. I suppose I was told that you prefer English to French. Am I correct?
Yeah. Yes. I think everyone study English. So
okay, you prefer English. Perfect. And then I would be really happy that I see your faces as well, and not just a little black spot or a nice static image. I know that maybe you do switch off the cameras for the connection, but it will make me happy to see your faces and you introduce yourselves as well. I would like to know, what is your name? I would like to know, what is your background? I would like to know if you have had any bioinformatics beforehand, and if you have done any programming, and I would like to know if you have done any immunoinformatics, or at least any immunology. Otherwise, welcome everyone to this class. We have Monday, Tuesday, Thursday, Friday this week. These mornings, well, they are mornings for France. And then we have Monday, Wednesday, Thursday, and Friday next week. There are not any actual exams to this class. However, there will be some projects. You'll have some homework to do every time. And either you'll be doing them individually or you'll be doing them in groups. And I would be asking you to hand in the homeworks. Is this clear? I think this is, because you're at the master level, I find it easier to work this way. It's not to penalize you with exams, but to encourage you to work and provide your own work at the end of the lectures. So before starting, I would like to point out 2 more little points that they might be of interest to you. There is currently an open call between Vietnam and France for students who would like to carry out a PhD in France. This is, as long as you are at the Master 2 level, it could be that some of you are interested in pursuing your studies further by carrying out a PhD. You might want to look into this possibility. I think the deadline is sometime in January. You need to figure out the details. You need to figure out, you need to find a host lab in France with a PhD topic. I think you will be given, from what I heard, 900 euros from this project in case your submission is honored. And then the French lab will have to contribute the rest for your PhD salary. In France, a PhD is 3 years long. At the end of the first year, you have the so-called CSI, which is like an evaluation committee, evaluates the progress of your work. You have, at the end of your second year, you have the evaluation of your work. And at the end of your third year, this is your thesis defense. Like you present your work at a small group. Most of the times it's open to the public as well. And then if things go well, then you get rewarded your PhD. What I advise you is don't go for a PhD if you're not sure you want to do a PhD. There are other opportunities. You can go for work. If you are interested, think why you would be interested in carrying out a PhD, first of all. And then secondly, try to see what is the topic that you are interested in and what is the lab that you're going to be going into. Make sure that you have good relations with the advisor, the PhD advisor, so it all goes smoothly. So that was one point I wanted to address. If you have any questions, please let me know on this one. And I have a second point. There is a second option for funding, which I think might be a bit more challenging because it's open to everyone and not only to Vietnamese students. There is the doctoral school, CBS2 in Montpellier, but there are doctoral schools everywhere in France, and they have their annual competitions. That is, they do a competition where you have to file your CV, your motivation letter, the PhD project that you will have decided with a potential advisor. Then you go through an interview, and at the end, they give some funding, some PhD funding. This PhD funding is good enough for 3 years to carry out your PhD. At some point in beginning of March 2023, these competitions will open up. So you could start looking from now to identify the different doctoral schools in France. And sometime in March, you could start like focusing on the ones you are more interested in. So these are the 2 points I wanted to bring up. I see that our colleague, our student, Tran An Xuan joined. Am I correct?
Yes, she is.
No.
Yes. Do you want to introduce yourself?
No, I'm just changing the device.
You just changed the device. So we don't have this person here. We don't have the Tran An Xuan. Am I correct?
Yes.
Okay. Well, nothing changed. Fair enough. So that was the 2 points. Do you have any questions on those? Right. If there are no questions on those, I'm sharing a PowerPoint. I could distribute these PowerPoint slides to all of you at the end of the class, or even now if you want to, but maybe at the end of the class. Several of my PowerPoint slides, they will be in French, but it doesn't really matter because I'll be speaking in English. So we are going to talk about immunoinformatics in our classes. Hold on a moment. How can I admit? There is someone who came in. And I have both I'm trying to manage both Skype and PowerPoint. I'll stop sharing to see how I can manage it a bit better. Right. I suppose you see my slides. I hope so. Right?
Yes.
Correct. Perfect. So we are going to cover 3 points today. We're going to talk about IMGT, the different databases and the different tools. We are going to talk about the immunoglobulins, their general structure, where they are located on the chromosomes, how is the synthesis of an immunoglobulin being generated at the molecular level, and then we're going to get to see the structure of 4 different types of genes. Genes, not the genes as I see there, genes in the sense of human genes. Right. So I start off with the first part, which is IMGT databases and software tools. Have a look at this slide, which is quite heavy. It's rich in information. IMGT, I repeat, is the International Immunogenetics Information System. Have a look at this slide carefully and let me know how many databases does this system have. If you look at it carefully, you do not need to know by heart. If you look at it carefully, you'll be able to find the correct number of the databases. Do you understand my question?
So you mean how many type of database the IMGT provide, right?
Correct.
Yeah, so I see we have 4 based on the color and the legend. We have the sequence, the gene, the 3D structure, and the last one I don't understand.
Right. So basically, correct answer, I was not asking only the type, I was asking how many, the number of the databases. And apparently you saw correctly, this is one database, all the cylinders are the databases. So we have 1, 2, 3, 4, 5, 6, 7 different databases. I wanted you to make the distinction between the cylinders and the rectangles. The rectangles are the different software tools that they are linked to the databases in order to analyze the information that is within the databases. We'll use a couple of these tools, and then you'll understand much better their utility. And as you correctly said, if you see this legend down here, there are databases and tools that they are dedicated to sequences. There is a database, I would even say there are two databases, because there is the GeneDB as well, and this one. But okay, these are databases. This is the database, the primary database that is dedicated to genes, the yellow color. This is structure databases, 2D and 3D structure DB, with the tools that they go along. And this is one of the most recent database, monoclonal antibody database, that we are going to carry a project on it. I say recent, however, it has been going on for more than 10 years. So it's rich in information. You do not need to learn anything by heart. The only reason why I show you this slide is that you understand the term knowledge base. What is the difference between a knowledge base and a database? A knowledge base is a whole system with several databases and tools and software tools. A database is a structure to store information. Therefore, I suppose you clearly understand that a knowledge base is much bigger than a database. IMGT is a knowledge base. It's a knowledge base, and I go to the next slide. It's a knowledge base in the adaptive immune system domain. This is the research domain of IMGT. So the question is, what is the adaptive immune system? For the ones who have had a bit of immunology, this is rather easy. So basically we have the immune system that has been divided into subsystems, we could say. There is the adaptive immune system, and which one is the other one? Do you know?
The innate immune system.
Exactly, the innate immune system. So we are going to focus in these classes, we are going to focus exclusively on the adaptive immune system. And for the ones who have not had much immunology, it does not really matter. Have a look at this picture, and if you take a mental picture, this is all you need to know to start this class. We are talking about vertebrates, and more precisely, we are talking about jawed vertebrates. Jaw is this part of the face, is this lower part, and the vertebrates that have got this jaw, in English it's written with J-A-U, W actually. This is jaw vertebrates. These are the species that they have the adaptive immune system. We are talking about 450 million years of evolution. And it goes from fish to human. So basically this word has been explained. And now look at the center of this PowerPoint slide. We have the B cells that they express immunoglobulins on their surface, and they're part of the antigen presenting cells. There are three different categories of cells. that they are called antigen presenting cells. One of them is the B cells, which through the major histocompatibility complex, they present a peptide to the T cell receptors of the T cells. And I have highlighted these three words: immunoglobulin, T cell receptor, major histocompatibility complex, because these are the three molecules that they are being studied by IMGT. During our classes, we are going to focus mainly on immunoglobulins and a bit on T cell receptors. Is this clear? I need a volunteer to let us know what is the take-home message of this slide, please. I can repeat it once more quickly, and then I would like a volunteer to explain with her own words. So basically, we are going to work with databases and tools of the IMGT knowledge base. IMGT focuses on the adaptive immune system. The adaptive immune system is found in jawed vertebrates, covering 450 million of years, going from fish to human. And it consists of the B cells that express immunoglobulins on their surface, and through the major histocompatibility complex, as they are antigen-presenting cells, show the peptide to the T cell receptors. Of course, this is an extra simplified slide. They're immunoglobulins that they are circulating in the blood as well. But for the purpose of the basics of immunology, for the ones who have never had any immunology background, this is enough to get us going. Is this okay? Do you have any questions on this slide? I'll focus on this molecule. This is the one that we are going to discuss in much more detail today. So if there are no questions, maybe we don't repeat as we said, and we'll go to the next slide. And you have a look at this one. Immunoglobulin membrane, IgM. I'll get to this later on. We have one heavy chain, you see it here, and we have one light chain. As a matter of fact, for each antibody, we have two heavy chains, you see them here, and we have two light chains. You see this red oval here and here? This is the so-called variable domain. I have a question for you. Look at the variable domain of the heavy chain and the light chain. What is the difference that you see? You don't need to know anything, you just look at this part, the variable domain of the light chain, the variable domain of the heavy chain. What is the difference?
Is the D region?
Excellent. There is this red, everyone could have seen that there is this red little line that is additional in this variable domain in comparison to the light chain. This part, the green part, is coded by a gene called variable gene, V gene. In the heavy chain of the immunoglobulins, there is this D for diversity. It's coded by the diversity gene. And this yellow is coded by a gene called J, joining gene, whereas the light chain of the immunoglobulin chains has got only a region coded by the variable gene and a region coded by the joining gene. Is this clear?
Yes.
Right. How are the two different chains, the light and the heavy chain, how are they connected together? How are they tied together?
The sulfate by the D-sulfate bonding.
Excellent. You see them here, right? As well as you see here, disulfide bridges, correct? Look at the constant region of the heavy chain. These are called the constant regions. How many do you see here?
4.
4. This 4 is not always the same number. Depending on the number of the constant regions, we have what we call different isotypes. IgM is one isotype. Do you know how many different isotypes do we have for the immunoglobulins?
5 isotypes.
Which ones are these ones, please?
IgM, IgG, IgE, IgA, and IgD.
Perfect. I would like you, the first little task for everybody, I would like you, how many are you all together? We are 14, 15. I would like, there are 5 different isotypes. I would like that you divide yourselves in 5 groups. I would like each group to take one isotype, and I would like each one of you to come back in 10, 15 minutes and present with 1 or 2 slides the different isotypes. Before we do this, I would like to share with you a web page as well. Maybe I stop this sharing, will facilitate my life. And I will show you something else. Do you see my screen? No, you don't, because I'm not sharing yet. Right. Now you should be able to see my screen, correct?
Yes.
Right. I have gone to the main page of IMGT. You see it here, www.imgt.org. It has got a little introduction. Here, there are the different databases. There are the different resources. We're mostly going to explore IMGT repertoire, but there are other pages that we're going to have a look at. Here, there are the tools. Maybe I should talk to you about IMGT education. The PowerPoint slides, and even more, like questions and answers, tutorials, you can find them here. If you are interested in finding something and you cannot really locate it because there are more than 20,000 web pages, you can put a name and search the web pages, IMGT web pages, as I did right now. I wanted to find the page that I have not located yet, that I want to show to you. It's the different isotypes. How can I, what could be the name? Isotypes, EMZT index is the one that we had a look at right now, and this is not the one I wanted. Excuse me. I'll be looking for the page I want to show to you, while you divide yourself in groups, 5 groups of 2 or 3 people, and each group will undertake one isotype. I would like that we come back in whenever you're ready, 5, 10 minutes, with one slide each, and one person of the group will present the features of its isotype. While you do this, I'll be looking to retrieve the isotype slide that I would like to show to you as well. Is this clear?
Yes.
Off you go.
Okay.
When are you coming back? How will I know that you're back? It's 9:33. How long do you need?
I think it's 10 minutes.
10 minutes? 9:45, we are all back?
Yes.
Perfect.
Sorry, Professor.
Yes.
The last person who is absent at the beginning of the lesson, she is in the meeting right now.
Right. Yeah. Sure. Thanks. I just saw your in the chat. I saw your message in the chat. Okay. Perfect. So please, what's your name? Juan, right? The person who was missing. Please get together with one group to prepare the homework. Well, it's not homework. It's the work during class about one of the isotypes. Okay? We are back at 9:45 with one presentation, one slide per group about the different features of the isotypes. I'll stay here, so if you have any questions, please ask me. In the meantime, I found what I wanted to share with you, and I put it in the chat. Please take advantage of this slide in order to describe as accurately as possible the different isotypes. And if you go to the bottom of this slide, you'll clearly see the graphical representation of the different isotypes. Maybe I share it with you.
A cascade of immune protein production that results in pathogen elimination. Okay, so here is the structure of IgG. It's made up of 4 peptides, 2 identical heavy chains, and 2 identical light chains. There are 4 IgG classes in humans from 1 to 4, and these subclasses are named in order of their abundance in serum. So here in the table, they show that the IgG1 is the most abundant in the human serum, and the IgG4 is the far less in serum. The difference between 4 subclasses, we can see that it is the hinge regions here. And the hinge region is the disulfide bond to connect the 2 heavy chains. So that is what we found about the IgG.
Nice. Thank you. That was nice. It was clear, synthetic, precise, and I'm happy that you saw the differences among the 4 different subclasses. Okay, thank you. So we heard about IgG. The monoclonal, have you heard about therapeutic monoclonal antibodies? Which isotype are they mostly? They're IgG. We'll get to see them later on. Okay, thank you. Who else is giving us another isotype, please?
So IgD is a monomeric antibody isotope that is expressed on the surface of B lymphocytes. It is also produced as a secreted form that can be found in the blood serum. It has the molecular weight of 170 to 180 kilodalton, and it is composed of 2 heavy chains of the delta class and 2 light chains. The function of IgD is not clear yet, and it is present in many species with a biological half-life of about 2 to 8 days. The IgD functions
Yes?
That's all. Sorry, my bad.
Okay, thank you. So I think we covered the 5 classes, correct? So, thank you.
Yes.
And now I'll continue. G. How many constant regions does it have on the heavy chain? 1, 2, 3. By default, we always start counting from the Fab, the antigen binding region, that is the Fab region, going to the end, that is, this is the CH1, and not this one. This is H1, this is CH2, and this is CH3 in the heavy chain. This is, I repeat, the variable region. This green, I repeat, is coded by the variable gene. This red thing here is coded by the diversity gene. The yellow is coded by the joining gene. This is the heavy chain, and this is the light chain. Light chain, there are 2 types. Could be either repeat the light chains of immunoglobulins. There is no region from the diversity gene coded. I have a question. Oh, this is in French it's charnière. In English, you said it really nicely. This is a hinge. The hinge gives a bit of flexibility to the molecule. This is the constant region of the light chain. We said it here. And this is once more the variable region of the light chain. Any questions, or is this clear to everybody? If you don't have any questions, I have a question. What are these 1, 2, 3, or here in the light chain, 1, 2, 3 little gray rectangulars that we see mostly on the variable region? Do you have any ideas? Any suggestions? Should I choose someone to answer? Or do I have a volunteer? If you don't understand my question, please ask me to repeat it. My question was the following one. I see 3 gray rectangulars there. 1, 2, and 3. What are these? Two of them, they are within the area coded by the variable gene. And one is covering the whole area coded by the diversity gene, as well as a little bit the 3' of the variable gene coded area, as well the 5' of the joining gene coded area. And what are these? What are they called? They are called CDR. They are the complementarity determining regions. And in essence, there are three loops that they are in charge of binding to the antigen. We called it the variable region for both the light and the heavy chain. This is the variable region. And these 3 rectangulars, they are the hypervariable regions. They're even more variable. And this number 3, exclusively in the heavy chain, is the one mostly in charge of the binding of the antibody to the antigen. Keep this in mind. CDR3 is the most variable among all the variable regions, and we'll get to see how it is generated on the molecular level. Is this clear, what we covered in this slide? Fine, so I can move to the next one. Have a look at this one. This is called space field. It is a 3D representation of an antibody. How many constant regions do I see? There are 3. It's an IgG. And how do you read this one? You see, for instance, variable heavy, constant heavy 1, constant heavy 2, constant heavy 3. This is the heavy chain. And you see it from this side as well, because there are 2 chains, the variable heavy, constant heavy 1, constant heavy 2, constant heavy 3. Right? And I suppose you see clearly, well, some people they have some difficulties with 3D, but if I point it out, I think it's obvious to everybody. This is the variable light chain, and this is the constant light chain. And from the other side, this light green color is the variable light region, and this red color is the constant light region. Is it clear for everybody? Please at least react.
Okay.
Yes? If you don't have any questions, I have a question then. What is this color here? There is this beige color. I need you to help me with this color. What is this? What could it be? What sort of molecule is an immunoglobulin? An immunoglobulin or antibody, there are two words that they're interchangeable. We can use either antibody or immunoglobulin, and they mean exactly the same. So my question is, what sort of molecule is this? Is this an RNA molecule? Is this a lipid? What is it? Lipid, not a lipid. Lipid, L-I-P-I-D. Lipid. Guys, what is an antibody? What sort of a molecule is? Is this an RNA? Is this DNA?
I don't think so.
Okay, so I agree with you. So what is it?
This protein.
It's a protein. Well done. It's not a trap. It's a simple question. I try to guide you to the correct answer of what this could be. This is a protein. The proteins, what sort of modifications can they have? I'm sure you all know.
It may be that some phosphorylation or methylation or acetylation.
Well done for trying to give an answer. However, this is not exactly what it is, but you're right. These are modifications, protein modifications. This is a glycan. This is a glycoprotein. Antibodies are glycoproteins. They have a sugar attached to them. And you might say, why is she insisting so much on this? This is extremely important. And I'll tell you why. Because we want to produce several times monoclonal therapeutic antibodies. If it's a glycoprotein, which system, biological system, do we need to use in order to produce these proteins? Can we use E. coli system, for instance?
No.
What do we use E. coli?
No, I don't think that we can use E. coli.
Why?
Because normally E. coli doesn't have the post translation modification. They can do the glycolate, like attaching a sugar residue to a protein. So I think E. coli is not a suitable expression system.
Perfect. Exactly what you're saying. Well done. So if we cannot use E. coli, what could we use to produce monoclonal antibodies or antibodies in general?
I think eukaryote cells or even animal cells.
Well done. We can use eukaryotic cells, or we can use insect cells as well to produce antibodies. And the reason is, as we said, because they're glycoproteins. So here, it's a glycan. There is a standardized way of naming the sugars that they are attached to an antibody. But we're not going to go into this detail. The take-home message of this PowerPoint slide is to remember that antibodies or immunoglobulins are glycoproteins. that consists of 2 heavy and 2 light chains. And I go to the last point that we are going to cover today, and I'll give you a homework. The last point, I request a bit your attention so you understand it. We get to see this map. We call it map, M-A-P, a map. Do you have an idea what we could be looking at? Do you have a suggestion? There is already written human IGH locus. Human, so basically we are a human species, IG, immunoglobulin, heavy locus. And we are on chromosome 14. How many chromosomes do we have? How many chromosomes do we have? The human species, how many chromosomes do we have? I'm sure you know, guys.
23.
Exactly, 23. And this is chromosome 14. Q32.33. And all of these, they are the immunoglobulin genes. Instead of giving you this static PowerPoint slide, I'll stop the sharing and I'll ask all of you, we'll go together at the IMGT webpage and I'll share my screen so we can all navigate together. Share screen. Do you see my screen?
Yes, it's on your screen.
Right. We start from the main page. The main page you can only get to by typing imgt.org. Please, let's get all together so we don't have to repeat 10 times and we don't lose people along the way either. Are you all there?
Yes.
You go further down, IMGT web resources. You see it here, I have my cursor. You click at IMGT repertoire. I click on this. I make it a bit bigger. Maybe I get this out of the way. And I go to the first one, locus and genes, and I click on chromosomal localizations. Right. The first question that I would like you to ask is to click, well, I haven't asked the question yet, but click please on IGH human. And we look at this. This is chromosome 14. By default, the whole scientific community has agreed to have the short arm of any chromosome at the top and the long arm of the chromosome is at the bottom. This, the short arm, is named P from the French word petit, which means short. And Q is the long arm because in the Latin alphabet, Q comes after P. You see, at the bottom of the long arm of this chromosome, there is the so-called locus of the IGH, the immunoglobulin heavy locus. It resides on the telomere of the 14th chromosome on the long arm. I suppose you know what the centromere and the telomere is. Have you heard there is this hypothesis that the telomeres, the human chromosome telomeres, they shrink a bit with age. And you all know, or you have heard, that the immune system of humans degrades with age as well. I mean, degrades. It's not as efficient as it is when we are young. In literature, there has been this suggestion that the weakness of our immune defense might correlate with the fact that the IgH locus is found on the telomere of the long arm. Is this clear? So now, I ask you the question that I would like you to answer. Please let me know which chromosome, on which chromosome we find the Ig lambda and the Ig kappa loci, locus loci in plural, of the human species. And share it with us and just present it to the class. How do you go about? Who can answer? Guys, are you there? Did you understand my question? I would like you to be a bit more reactive, please.
Can you repeat the question?
I repeat the question. Excuse me. I just showed to you how to retrieve the location of the immunoglobulin heavy locus. And my question is, on which chromosome is the Ig lambda and Ig kappa loci found? Yes? Anyone? I would like someone to repeat what I just did for different locus, for different locus. And more specifically, I can repeat once more what I did.
You asked us to
I asked you, yes,
what you just did, right? The
Correct.
navigation from the main page.
Exactly.
So
Can you do it?
yes.
Do it for us, please.
So from the main page at imgt.org, we move to the section called IMGT matrix source and click on.
Yeah, but do it. Don't just put it. Share your screen, please, with us
Okay.
and show it to us.
Can you see my screen?
Yes, now I can.
Okay, so I go to the IGT web resource. I click on the IGT web platform.
Yes.
Then I move to locus and gene. Click on
Yes.
the chromosomal localization.
Correct.
Then here I move to this section. I click on IGH human.
No, IGH, I showed it to you. I asked you which chromosome do we find IG lambda and IG kappa. So go back one page,
Yes.
please.
I try to
Yes. No.
IG lambda.
IG kappa, IG lambda. You choose one of them. Which one? You want?
I think I'm gonna choose IG lambda.
Perfect. Go
This one,
ahead.
yes.
Yes. And describe it to us.
This IG lambda.
Locus. This locus. Yes.
This locus. It's focused on the this position 11, 12.
Which chromosome is it on?
It's on the chromosome 22.
Which arm is this on?
It's on the long arm at brand 22Q11 2.
Perfect. And now look at this. It tells you the locus is forward or reverse position. Do you understand this? It's described in the text above the representation, and it says that it's forward. It's in bold. Why is it forward? Because the gene order in the IG lambda locus goes from 5' to 3'. That is in the same direction with the chromosome direction. Is this clear, Miss, as well?
Yes.
And please, I have one question. How many variable genes do we find on the IG lambda locus according to this schematic representation? How many variable genes do we find on this locus, on the IG lambda human locus?
Based on the schematic, there are 3 variants.
How many variable genes? You are close, but you have not given. I wait for a number. How many variable genes? That is immunoglobulin, light, variable genes. Where do you see the immunoglobulin lambda variable genes?
I see it from the
Yes.
blue arrow.
And how many are there variable genes?
I think there are 3 different variants.
73 and 74.
Exactly. There are 73 to 74, that's what's written there. How many immunoglobulin lambda joining genes do you think they are there?
They're joining, there are 7 to 11.
Be a bit more careful, please. Joining,
This one.
there is one. How many constant there is there?
So 1.
Why is there the 7 till 11? Because they are repeated. There are parentheses. There is G-say, like joining constant gene. There are 2 together. There is another 2 together. There is another 2 together. There are 7 till 11 repetitions. Okay?
Okay.
How long is this area? How many kilobases?
Then 1050 kilobases.
Perfect. Thank you. So that's nice. I'll go and share my screen once more. No, I don't want to share my PowerPoint slide. I would like to share once more my web browser. I start off from the main page, as if I did not know much about this site, and I go to IMGT repertoire. This time, I'm gonna go to the second one, locus representations. There are several different species. Remember we said from fish to human, vertebrates, jawed vertebrates. I'm gonna take immunoglobulin lambda human. And I have this map. I start off with this one, and actually I go further down for the IG lambda joining genes. How many do you see here? 7 joining genes. And this is the JC cluster. If I make it a bit bigger, you see what we discussed about? The J, the yellowish, with the constant, and there is JC 7 times. Is this clearer, what we saw the chromosomal localization? Right? And I go to the very top. This is the map. These are all the variable genes. I can tell you what they are. I think you will forget, you won't understand, it will be difficult, and this and that. However, if I ask the task that you have another half an hour to complete, and you'll present it tomorrow in the morning, we'll start with this one. This will be your homework. I ask you to understand what you see on this map and describe it to me. However, you are several people. I don't want every single one to take this map. I go back to this one. There are locus representations of immunoglobulin heavy, immunoglobulin gamma, kappa, immunoglobulin lambda locus. And then we have 4 more loci: T-cell receptor alpha, T-cell receptor beta, T-cell receptor gamma, T-cell receptor delta. I would like each one of you, there are more than 15 loci that they are available here, to take one of these loci, describe it to us, what you have understood, and compare it to the human one. Like for instance, I show you once more, this is the human IG lambda. If I decide to work on the bovine IG lambda, bovine is the cow. This is the loci, the locus. Are they identical? Are they different? And what are the differences? I'll help you a bit more. In case you find it extremely difficult, you say, what is she talking about? I don't understand anything. There are links in the page that they can take you to understand how to interpret this picture. If you are the sort of person who prefers reading, if you go to references and news, at the main page of IMGT, you click here, you go to IMGT references, you click here, and there are several publications. You can choose one publication that you feel, I'll let you choose the publication. I won't give you the publication. These are all IMGT publications. You can choose the relevant publication that will allow you to understand how to interpret this map. Is this clear?
Okay.
What I'm asking for you?
Yes.
I'll give you something more. I got IMGT posters. I want I would like to show you this poster, right? Do you see all?
Yes.
So we are looking at this left side, the blue background. This is what we discussed about today. Vertebrates from fish to human. And eventually, from the published databases, we are going to extract the DNA, and that's what we are looking at right now. We looked, for instance, at chromosome 14, and we said that at the telomere, there was the immunoglobulin-heavy locus. Your classmate had a look at the immunoglobulin lambda locus of the human, and he told us that we are at chromosome 22. I told you that there are in total 7 different loci per species, and I asked you to go and understand the map, how we read and how we interpret these maps that they have been created by IMGT biocurators. Biocurators are people who take this DNA and annotate it. They do this sort of work. They are experts. They take the nucleotide sequences, and with the help, well, here is a pencil or a pen, but there is the help of software tools, they identify the different variables, the diversity, the joining, and the constant genes. With the help of a so-called ontology, they identify these genes, they describe them, they classify them, and they put them in a specific database, which is IMGT/LIGMDB, and when the annotation has completely finished, they put them into this IMGT/GeneDB. Right? So I repeat your task for the rest of today. I'll let you the other half an hour. And if you need more time, you'll do it at home and you'll present it tomorrow. So tomorrow, I would like each one of you, I repeat, to have chosen one locus from one species. You decide the species, you decide the locus, either immunoglobulin locus or T-cell receptor locus. You go through either publications or the web pages to understand. I'll show it once more to you, to understand what we mean by the different symbols and the different names we see on the map. For instance, I show you this. Here, I don't see it that clearly, but it doesn't matter. I see, for instance, 4-59, and then I see 1-58. What are these names? How are they genes? Why do they have different colors? What do the different colors mean? How are they named? Why is this 4-59 and why is this 3-43 or 48? Here I see, if I go, if I go further down, I see J 1-5. Here I see C. What sort of genes are they being shown? Here I see kilobases. Here I see this little gap. What does it mean? I see some arrows that they go. Here I see 5', 3', and I see an arrow going this way. And then I don't see an arrow. I see at the previous slide, what do I see? I see a triangle here. Do you see? Or I see a purple here. And this is a bit different to the other genes. This is GL something. What does it mean? I see another purple here. I see this triangular coming out here. How do I interpret all these things? So you choose one locus. Please make sure you coordinate among yourself that each one of you has got a different locus. Okay?
And I want to ask that each of us have how many times for each locus?
Say this again, repeat your question.
So I want to add the time for each person to present our locus we choose. So
actually, it does not become too repetitive. You can work in pairs. You can be two people for one locus. So you can help each other as well. And the time is up to you. Enough to describe the locus. However, please, I repeat, coordinate among yourselves. So we hear 7, 8 different loci. So we do not hear the same loci. Okay? Is this clear or do you have any questions on it? Do you want more help? Should I give you more hints? Guys, do you know what I'm asking, or are you gonna come back tomorrow without having done the homework?
I think it's clear for me.
It's clear for you, great. Do you want me to put on 1 or 2 different articles that can help you with this, or do you think you'll be able to find them?
I think you can
Could you share with us?
I'll give you just one. I'm trying to find the chat now. Chat. Okay. I gave you the DOI. Is this okay? For instance, we are into looking in more details of its locus, and this is your task for tomorrow. Okay?
Okay.
So thank you very much, and I'll see you. You can stay working now, and I'll see you tomorrow, 8:30 or so. Again, is this okay?
Yes. Okay. See you tomorrow.
Yes. See you tomorrow.
Thank you all.